Cells have to do lots of work just to survive. And we know that the all-important molecule that powers most of that work is ATP. Now, cells generate ATP by breaking down organic molecules. Basically, the energy that is stored in the bonds that hold the organic molecules together is used to build ATP. Now, most cells do this through the process of cellular respiration, which takes place over a series of stages. And in our previous videos, we looked at the first two stages, glycolysis and pyruvate oxidation and the citric acid cycle. One of the things that we took away from those videos is that the energy payoff, at least in terms of immediately usable ATP, is in fact pretty meager. If you want to be specific, it's 4 ATP. We got 2 from glycolysis and we got 2 from the citric acid cycle. The thing is, most of that potential energy from that glucose molecule is at this point has been transferred and is being carried in the electrons of NADH, of which we have 10, and FADH, of which we have 2. And to get at that energy, to make use of it, what we have to move on to is the final stage of cellular respiration, the electron transport chain, and oxidative phosphorylation. In a eukaryote, this final stage of cellular respiration takes place in the mitochondria. It actually takes place in what are called crista. These are sac-like structures on the inner mitochondrial membrane, and it relies upon a series of protein complexes embedded within that membrane. In fact, these protein complexes are what we refer to when we talk about the electron transport chain. Now, as it turns out, there are 4 of them, and for convenience, we just simply label them numbers 1, 2, 3, and 4. But it's important to appreciate that they are functionally linked by electron shuttles, which are able to transfer electrons from one complex to another. So let's try and put all this together. How do we go from NADH and FADH, which are carrying electrons stripped away from glucose, and put that together with this series of protein complexes that we call the electron transport chain in the inner mitochondrial membrane to get our big payoff of ATP? Well, that's really the big question now, isn't it? So let's buckle up and take a look. This all begins when electrons from NADH are transferred to complex 1. Now, that means this is a redox reaction. In other words, NADH is oxidized. It's actually oxidized back to NAD+. It means that complex 1 is therefore reduced. And what follows is that those electrons are then transferred through a series of redox reactions down through complex 1 until they are transferred to the electron shuttle. And the electron shuttle actually takes them over to complex 3. Now, what happens at complex 1 and what happens at complex 3 as these electrons are being moved is really pretty cool, because each time these electrons move on, a little bit of energy is released. And complex 1 and complex 3 use those little bits of energy to move protons. What they actually do is they move protons from the mitochondrial matrix into what we call the intermembrane space, the small gap between the inner and the outer mitochondrial membranes. Now, hold that thought for a second, because what about FADH2? Well, FADH2 brings its electrons to complex 2. They are transferred again via a redox reaction, meaning FADH2 is oxidized and complex 2 is reduced. Well, complex 2 then passes those electrons onto the shuttle, and the shuttle takes the electrons onto complex 3. Now, what will follow is that, again, through a series of redox reactions, they will move through complex 3, they will get picked up by the second shuttle, and they will get delivered to complex 4, and they will then move through a series of redox reactions through complex 4. And complex 4 is going to do the same thing that complex 3 and complex 1 did. As those electrons are being moved, as they give off small amounts of energy, that energy is going to be used to pump protons. So let's just hang on here. We've moved a bunch of electrons from NADH and FADH through complexes 1 and 2 to 3 to 4, and as a result we've pumped a bunch of protons into this intermembrane space, but still not seeing ATP. Here's the really cool thing. The pumping of those protons as a consequence of the movement of those electrons has created a proton gradient. There is a much higher concentration of protons between those two membranes than there is in the matrix. And what that generates is what we call a proton motive force. Those protons want to move from the area of high concentration to the area of low concentration, but there's a membrane in the way. And they can't do that because the properties of the membrane, the lipid bilayer, mean that they can't just passively diffuse through. But embedded in that inner mitochondrial membrane is a large protein complex, and that protein complex serves as a gateway, a passageway for protons to move from that high concentration back into the matrix. And that protein complex we call the ATP synthase. Its name tells you what it does. It synthesizes ATP. Now, this is a pretty complex process, but to give you an idea, down the middle of this protein complex, it's kind of like a barrel. That's the gateway, that's the passageway. And as the protons move through this barrel, as they move through the passage, it causes another part of the complex to turn. Think of it like a cog. And as the protons move through and the cog turns, it activates catalytic domains. And what these catalytic domains do is take ADP and free phosphate and bring them together to form ATP. So in other words, it's actually the energy from that proton gradient generated by the movement of the electrons that actually drives the production of ATP. Fantastic. And if you add what comes out of this process of the electron transport chain, oxidative phosphorylation, with what we got from glycolysis and pyruvate oxidation, the citric acid cycle, you get per glucose about 30 ATP. What a magnificent process. But just hold on a minute here, because there's one whole aspect of this cellular respiration equation that we haven't mentioned since the very start of the glycolysis video, and that's oxygen. Remember how I started off that video talking about why do you breathe, and oh, you have to have oxygen because it's indispensable, blah, 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 etc., etc. And then since then, nothing. Well, it's actually now. Now at the end of this final stage of cellular respiration, if you want to be specific, right at the end of the electron transport chain, that oxygen becomes so important. Because those electrons that started in glucose, that moved through NADH and FADH and then through the electron transport chain to the end of complex 4, are now transferred to oxygen. Oxygen receives electrons from complex 4, it takes protons, and in so doing, becomes reduced to water. That's right. The oxygen that you breathe ultimately gets reduced to water. This is the ultimate importance of oxygen here. If you have no oxygen, complex 4 has nothing to transfer its electrons to, which means it can't take electrons from complex 3, which can't take electrons from complexes 1 and 2, which can't take electrons from NADH and FADH, and the whole electron transport chain grinds to a halt. So if cells have no oxygen, then there's no functioning electron transport chain, there's no proton gradient, and there's no ATP synthesis. No ATP means no cellular work, and that's really bad news for a cell. So really, the story of cellular respiration is the story of electrons moving from glucose to oxygen. That is ultimately how cells produce that ATP. What an absolutely beautiful piece of biology. All there's left to do now is sit back and admire it. So just relax and, as I've told you, breathe.